I've been working on a project sponsored by GSK, the pharmaceuticals company, and EPSRC. This project brings about lots of different technologies involved in biological research. But today I will talk about one of those technology, which is ChIP-seq, and we will show how this result can be improved using Markov random field model. So I will start my presentation giving a little bit of background on biology so that it would be easier for you to understand what I'm trying to do. So genes in our body actually produces proteins, at different times and locations, so that for our normal biological development process to go on. But not all genes are active all the time. For these to be active and producing proteins, there are other factors that work together to make the gene regulated. And one of these factors are some chemical modifications that happens around those genes, and also some proteins and transcription factors that bind at different location of proteins, genes, to make those genes active so that it can produce the right amount of protein that we need and when we need it. But this process is very complicated, and also it is related to various diseases. So if the process is disrupted unduly, it could cause even life-threatening diseases like cancer and other diseases. So it's very important for biologists to know where these regulatory things, like proteins and chemical modifications, are occurring around genes to regulate this protein producing mechanism. So there are different technologies that are used for finding out the location of these proteins, and one of them is ChIP-seq, which is chromatin immunoprecipitation technique followed by next generation sequencing, which we will talk about today. So with this ChIP-seq technology, what happens is with specific antibody and immunoprecipitation technique, a specific protein-bound DNA sequences are filtered out from any cell or tissue, and then this is reversed, and only the DNA sequences that are bound by that protein of interest are filtered out and used as a raw sequence in the ChIP-seq data. Later, these DNA sequences are mapped back, which is number 6, mapped back to the whole genome, and see where these DNA sequences which are bound by the proteins come from. And using different statistical analysis, we find out which of these locations are significantly bound by that protein, so we can see these protein come from that genomic location. ChIP-seq data is very complicated data, as you can understand. There are like lots of pre-processing of the data, and also there are technical errors and involved. So we will talk about two of those complex characteristics of ChIP-seq data today, which we try to improve. One of them is background noise. So basically, in the technology, when you are collecting the sequences which are supposed to be protein bound, there are some random sequences which are also filtered out by the technology, which actually creates a noise. So we should actually ignore those noise to get the actual location of the protein bound genomic location. And another thing is spatial dependency, which is when we get the as you can understand, our genome is like 1 million base pairs long. So in statistical analysis, what most of the methods does is divide the whole genome into small small bins or windows which are fixed length, and then summarize the counts of sequences around those around those bins. So how many counts come from like each location, and then they statistically find out which are the counts are significantly enriched enough to be called as protein bound. But when we divide the whole genome into small windows, it actually introduces spatial dependency, because some of the protein bound locations actually cross between windows. So for example, for one protein, the peak might show you a very sharp peak. So in one of the bean, it shows very high count of protein. So you immediately say this is protein bound. But for some proteins, it will go around some regions. So they create that sort of like broad peaks around like lots of regions. So each beans or windows have small counts, but at the same time, because it's broad, it will have like broad regions have small counts, and those regions should be called protein bound. But if the statistical analysis method that you are using does not actually consider this spatial dependency, you actually ignore lots of useful information to model the data. To address this issue, we have adapted random field model to model our ChIP-seq data. So basically how it works, first of all, a general idea of how you see whether a bean is enriched by the protein. So in that analysis, so you want to know, so for example, a mth bean, whether this is enriched by the protein of interest or not, which is, say, a latent variable called x. So you want to know whether x is 1, which is enriched, or x is 0, which is not enriched. So if y is the count, the probability of that bean to be enriched will depend on whether that count comes from the actual signal component or the background noise component. And that theta s and theta b is the signal and background densities of the whole data. But in random field model, it assumes that every bean, the binding profile actually meets the 1D Markov properties. So in that case, for each bean to be called enriched, it also look at the adjacent beans. So for example, for mth bean, it also look at m minus 1 bean and m plus 1 bean as well. So it incorporated into the algorithm so that each probability will then look into the probability of the adjacent beans. So this is how the spatial dependency are taken into into considered in the model. So we have analyzed our data with random field model and also with a latent mixture model, which does not take this spatial dependency into account. And for the time constriction, I didn't actually describe what my data is. The conditions are actually the biological condition that data came from. So it's a time series ChIP-seq data, which are collected at 3 time points, which is 0 hour, 1 hour, and 4 hour. And we have got 2 biological conditions. One is stimulated with some LPS, which is considered as a control data here. And because the data came from a pharmaceutical company, they used a drug to look at the difference between 2 data sets. So this is defined as drug in the condition. So if you could look at the numbers of the MRF, and this is for just one chromosome for H3, the histone protein. In each case, in each biological condition, MRF has produced a lot more, around 30% more, regions that are bound than the mixture model that just using a negative binomial distribution. To give you a visual how these 2 results are different, we have given you the integrated genomic view of the count data. So if you look at these blue boxes, you could see there is a region. So if you could say among the million base pairs, say this is a just 1,000 base pair area, or maybe 2,000 base pair area, and this is divided into 200 base pair area in our model. So in each window, say we have a very small counts. This whole area is ignored by the negative binomial distribution, but resulted in our MRF model as bound by the H3 protein, just because even the counts are very small, it has small counts around the area. So there must be something happening in that genomic location to be for these whole lot of counts to be, or DNA sequences to be mapped there. So if we in the MRF model, because all the counts are considered separately, they will be ignored because the counts are very small. So to summarize the result, we have seen the MRF model, the Markov random field model, produces more bound regions than the mixture model, which is just using negative binomial distribution at the same false discovery rate. With MRF model, we can identify these broad regions rather than sharp peaks of the counts. And also, if spatial dependency is not accounted for in the model, many useful information about the protein binding location may be discarded by the models, which is very when you were comparing between data sets, it could be detrimental for the result. Thank you. So I thank my funding institutions and my supervisors.